Okay, so we'll get started with the webinar today. I'm Emma Ganley, Director of Strategic Initiatives with Protocols.io, standing in for Julia Kurtz, who couldn't make the webinar today. But we'll give a nice introduction to Protocols.io and a little bit of a live demo, assuming time goes in our favor. So a few little housekeeping notes. We are recording the webinar. I just started that, so it's already in being recorded. Anyone who has registered will receive a link to the recording and to the slides afterwards. So you can watch back or check the slides, and people who registered who didn't make it will also get that. If English is not your native language, closed captioning or you have other accessibility concerns, closed captioning is enabled. So if you want to have those running, you can set that up. There should be time for questions at the end of the webinar. You can either pop them in the Q&A or the chat. I probably will not be able to see them while I'm presenting, but I'll take a look at the end and go through things. And then just at the bottom here, pointer that you've obviously discovered this webinar somewhere, but we list regularly other webinars and we have different ones. This is that very first introduction. There's a slightly more advanced one. And there's also one more about how to obtain credit for publishing and using protocols.io. So if they're of interest, you can find them there. If you would like a more tailored session with one of the protocols.io team, there is a place where you can sign up for a one-to-one demo, or if there's a couple of you using the platform, you can join together and connect with one of us. You can select a time from a link here and choose a time that will work for you to connect with one of our team. But here's where we'll go today. I'll introduce protocols.io, talk a little bit about why method sharing matters and about modular publishing today and what the difference is really between the protocols, maybe data code and the papers and so on. And we'll cover a bit of an introduction on the platform, showing you a few key pieces of functionality. So protocols.io, if you are not familiar at all, is a workflow solution specialized for protocols and methods. So the ideal way to use it is throughout your research process to capture details of what you're working on as you develop a method and get it to the point where it's the method that you will use and you are using for some kind of research question. You can also publish with the tool, so it has a lot of public content on it as well. We have academic customers and government and corporate customers. Can also be used as a free tool. There's a freemium version which has some limitations on some of the functionality, but will allow you to create a couple of private protocols and publish them, make them publicly available. So in terms of use of the platform, you can see we have a lot of users and we have a bit over 25,000 published protocols on the platform now, and a lot more private protocols. And this is where customers or other individual users are creating content privately and perhaps choosing either not to publish at all, or they've got iterations and versions before they're ready to publish, or they're still developing and might publish at a later date. Or maybe it's, just buffer solution recipes which don't need to be published necessarily. The mission of protocols.io is very simple. We want to make it easy to share method details before, during, and after publication. So throughout entire research workflow, all of that publicly available content is open access. So it's all available with a CC BY license. That means that if there's content there that is valuable or useful for you as a researcher, you can actually create your own version of that protocol. We call it a fork. So you can fork any of those public protocols to make a version for yourself that you can amend for how you would be able to use it depending on your circumstances. So you can adapt it and make changes and reuse very easily. As I've just said, there's a huge part which is the open access, but then this private side, this is much more what is and for the paid version really, because you have a limit on the number of private files with the free version, which allows you to use it and do most things, but it does limit the collaboration capacity that you can have on the platform. So the private side is about with your team, perhaps having a common workspace where you're all members, but you can collaborate and keep a common store of all the protocols that you are using within that team. If they're all public, then that works more easily. But if you want to keep things private, that side is the part that becomes more kind of supporting the business model for the platform. Um, you can check if your organization already has premium protocols.io. Some of you mentioned you're all across Europe. We have some license holders across Europe. And if you visit this page, plans/academia, or the protocols.io for institutions, partway down the page, there's a drop down which will show you the organizations that do have access. So you can see if your organization is there. If they are listed, then if you create an account generally using your organization email, it will automatically be premium. That would mean you wouldn't have any of those limitations I talked about. If it's not there, you can still make that free account. This is the open research account, and do all the things essentially to make content available. So we're very much supporting that open research side with the platform. And why is it important to make methods available? So this is a key thing for reproducibility, which is something that has been flagged a lot in recent years. A lot of concerns about whether, excuse me, my mouse is making the slides flick backwards and forwards, whether research that has been published can be reproduced. Can someone else read your paper and actually understand what you did and replicate the methods, get sufficient information about the methods? And what we see, these are tweets, but we see the same thing now on other platforms like Blue Sky. What we often see is that researchers are getting stuck looking through papers for details of methods, and there are not enough details in those papers for the authors, for the researchers to be able to understand what the authors originally did, to be able to replicate and reproduce. So that means those papers that have been published have limited value in a way because we can't necessarily build upon. It's hard to reinterpret data if we don't have all of the methods available. This was emphasized a lot with an initiative that was undertaken a little while back now about cancer papers, published perceived important cancer research papers, where an initiative was undertaken to try to replicate and reproduce the results of those papers, 53 papers in total. And could we actually get the information about the experiments that were performed and reproduce them? The short answer to this question is no, it was not possible to do this. What happened was in trying to undertake the initiative, the researchers originally looked at the papers, and there was generally not enough information in the papers. As you can see on the right-hand side, 0% of protocols were completely described. But they also talked to the authors, and the authors of those papers, the original papers, often didn't have the information either because perhaps postdocs had left the lab, people had moved on, or a laptop hard drive had failed, and they also didn't have enough information to reproduce some of the experiments. So this initiative ended up being stopped after they tried to reproduce, I think, 18 of the 50 papers because it essentially wasn't possible. It was too expensive, both time and money wise, and they realized they could not do this. This is very similar still today. That previous study was sort of from that post in the Atlantic was from 2017. This is from just this year, earlier this year, a big project in Brazil looking at reproducibility of methods. And very similarly, there are weaknesses in the way we produce, document, and communicate science, and that there's very low levels of reproducibility available from the data that's being shared. So we need to do better, and that is where we start to see change happening. We start to see, I mentioned modular publishing. This is this way of being able to say my paper in the middle here is kind of one key piece of my research, but actually there are many other parts that are distinct modular outputs. So it may be the method, it may be some particular analyses, it may be some structures, it may be some code or images or movies. Microscopy, data, etc. It can be all of these things. And we are now seeing more and more kind of interconnected infrastructure where the different pieces can be placed. So we are seeing some nice examples. So this is a paper from Nature Communications, and you can see that in the data availability here, they've talked about specific materials being deposited at AdGene, protocols being added on protocols.io, and specific data on the protein data bank. So This is really good example of what we would like to see researchers doing in general to make sure their research is very clear, all the details are available of all the different component parts. And if you do this, it's really nice too, because actually those component parts might have been created or developed by a different author. So if we look at that particular example, the first author on the paper was Elizabeth Bell, and down the list, fourth author was Kelsey Ramirez. And if we look at the two protocols that were mentioned in that paper that have been published on protocols.io, Kelsey Ramirez was clearly the expert who developed the methods, and so by pulling the methods separate from the paper, we can make sure that Kelsey Ramirez is understood to be the expert for the method here, and she gets the recognition for the development of the protocols on protocols.io, while Elizabeth Bell applied the method and performed some experiments with them. We can understand better who deserves credit for which part. And it's very interesting when you look at that, how protocols appear and papers. Sometimes the visibility of the paper is very different from the visibility of the protocol. So this is a different example, but also a Nature Communications paper. When you look at the protocol on protocols.io, there's always a metrics tab. And when you click onto the metrics tab, you can see how accessed your protocol has been. And in this instance, there's been almost 130,000 views of the protocol. And if you actually compare that to the paper that's published in Nature Communications, interestingly, that's at about 37,000. So there are a lot more views of the protocol than there are the paper. So sometimes, the perceived impact of the method may be much higher than the paper in terms of ongoing reuse and utility. And so that brings me to protocols.io. This is a place to capture dynamic step-by-step methods on the platform. And this is one example which I'll show you on the platform in a moment, but there's various things quickly to point out. Can have different versions, can be linked with a paper. Although we were acquired by Springer Nature, so we're a Springer Nature tool now, we're very independent, and it's kind of a modular tool, so it can be linked with any publisher, any journal. So eLife is not a Springer Nature paper, but we can add the citations to make sure that there's this nice interlinking between method and publication. The protocols will get a DOI on protocols.io. You can see that here, so that they can be independently cited. We have a lovely measure of reproducibility, which is this thumbs up. And that's not just I like it, that's actually I managed to reuse this protocol. So if we had had protocols like this for all of those cancer biology papers mentioned earlier, it's possible that the projects would have been reproducible. When you scroll down on the page, it has a lovely template format to capture any kind of step-by-step method. And people can ask questions and put comments on the protocol. This works both on public and private protocols. So if you're still developing with your colleagues, you can have a back and forth about optimization suggestions or get feedback on certain steps from your boss in the lab or wherever, and have a back and forth. You can clean those off if you publish it, a bit like a Word document. You can resolve all the comments, accept everything. And then when it's public, comments come on and they're publicly available. So that means you almost have a public FAQ on the protocol, really, which allows you to see if someone else has asked a question, gives you a lot of troubleshooting advice, things to try, but very specifically on different steps of the protocol. So this example has a lot. I don't know if you can see, but up there it says 14,000 comments. That's atypical, that's much higher than general, but it's very useful functionality, which allows for the protocol and an interaction with the authors of the protocol far beyond what you would have with a paper. And this publishing process in general, it's so easy to add a link to the protocol on protocols.io or create a collection of protocols on protocols.io and link to them from a paper. So I'll very quickly show kind of the publish process in a moment, but you'll get the DOI for the protocol, in this instance, collection. And in the materials and methods of a paper, you can link to it very, very easily and just say comprehensive details of the method or methods are available on protocols.io and pop the DOI in. And you can add a citation to the protocol as well in the reference list. So you have it in both places, and it's very trivial and easy to do. what's very nice about this is that if you spot a mistake or you subsequently optimize the method on protocols.io, you can update on protocols.io in a new version. And if someone clicks from the paper, they will still always land on that version that was made public when the paper was published, in this instance, version 1. But if a newer version's been created, you'll see that very prominently, and a viewer, a reader can have access to the newer version very easily, and they'll be able to compare and see what's been updated between them if they want to. This is the publish process. If when you're creating a protocol on protocols.io, you'll see very prominently when you're in the platform a button to publish. Reserved UI is very similar, but it will not make it public immediately. It will simply tell you what the DOI will be. So if you want to coordinate with publication of a paper, you can know the DOI, put that into your paper, get a private link for reviewer and editors to have access if they want, but not fully publish it, or publish will take it straight through. And that process is quite easy. It's these steps that you see here. Um, and it will require you to fill in some key metadata, but otherwise it will just go through. It would probably no more than 5 minutes if you have all the information to hand to just click, click, click and make sure you've added all the authors, any funders, things like this. And then it will be published at the end. We do some screening, so there may be a delay for a screening step, but it's very easy to do. And this is really nice because it means that you're creating a discoverability for your methods that's separate from a research paper. So in this instance, this is a nice highlight for this. Someone on Twitter was looking for a protocol for RNA extraction on primary cortical neurons and asking if anybody had any suggestions. And that was a PhD student or someone in Chile, I think, a postdoc in I think UCSF, Elena there replied and suggested this method on protocols.io, very clearly an RNA extraction protocol. What's interesting is that the protocol was linked with a paper from GigaScience. So it was the protocol to go with that paper that was published. And when we look at that paper, it turns out the paper was a method and an analysis of three-spine stickleback parasite, so a fish parasite. So it's very, very unlikely that you would have found that when looking for something on primary cortical neurons, that you would have found a paper on a fish parasite. But the fact that the method existed separately meant that that was findable and discoverable much more easily as a separate research output. So we're going to jump in a moment into actually showing you the platform, but you have a workspace. This is like a file manager. Essentially, it's a place, but it can have multiple members. So in the workspace, you have an administrator. You can add people in there, the whole team or department, or just per project. And it can either be public or private, and you can control how people join. The file manager is where all the content in that workspace is kept. So this is just a centralized secure location. The people you give access to will be able to see what's in the file manager. That's the members of the workspace. But otherwise, it's a location you can create some structure in there, add the files, create protocols, and so on. When you make a protocol, you'll be using the editor. So that is the third view that you have is the editor, and that has that template format to create a nice structured protocol. Right, okay, so you might have to bear with me while I stop the share and then share again a different window. I'll have a quick check about the questions. While I do that, I have a question. If a protocol is published in a later version is done with the DOI. If you're referencing the last version. Okay, the DOI is different. So if you make a new version publicly available, then it is each version will have its own DOI. So you would land on the version that's linked to from the paper. Sorry, I'm playing with the things on my screen to try and find the right window before while I'm talking to you. You would land on the version that was linked in the paper, but the new version would have a separate DOI, but they're always linked to one another. Hopefully that answers that question that was in there. Right, let me share a different window. And I can show you a little bit of the platform and how it works. So I mentioned earlier that I would show the protocol that I had on the screen before. That's this one here. I kind of talked about the versions just now as well. You can see that they're all here. And this one has a DOI that ends 2VN. And if I click to an earlier version, that's got a different DOI. So that's exactly what I was just talking about before. But we'll go back. And on the right-hand side, you can compare here any of those versions to a different version. So the one that you're on, you can compare either of the later or the earlier version to that protocol very easily. The eLife link, that's clickable through, excuse me, table of contents is generated from the sections during the steps. So you can see you've got different color coding, and those different sections allow you to navigate, but also to separate out into parts, the different parts of the experimental process you might be following. So if some parts are, on day one and then on day two, you need to jump down here, you can just jump straight back to there where you need to be in the platform. What else on here? The comments are all up here, but if we scroll down, you'll see them on the individual steps. So we can have a look at what's on any of those, and you see the question and answer from the authors. So you also have a mechanism to message authors if you're on here. We've added some translational functionality. So all of our published protocols are now available using AI in multiple different languages, these 5 here. So I know we've got a few different representatives from different European countries, maybe Africa, but I'm not sure. I've lost track if there was anyone from Asia here today, maybe not the best time. But if you want to look at a version in a different language, then you can. And anecdotally, we've heard that works quite well. If you have feedback, if you look at that, let us know what you think. If you're making private files and you have premium protocols.io, you can work in a different language and translate into English as well. So there is a lot of nice functionality for that. If I click into the file manager, you can see I've got the ability to manage translations over here. So if you click here, I could create translations for this protocol. And do things like that. Where should we go? We were in here. We also say we have copy fork. So if I found a public protocol like this, and I think I really want to use this protocol, but I would have a slightly different setup in my lab, then I can make a fork of it. I can only make a copy if it's my own. So that allows you to make templates for your group or your team where you can copy, the basics across. Fork is the only thing I can do for someone else's protocol. So I can make a fork of it, and that will create a private copy I would put in my workspace, and then I can make changes to it and do what I want to. Run is very nice functionality that allows you to actually step through all of the steps in a protocol. So if I say start run here, it's going to create a run record in my workspace, which will allow me to step through all the steps. So if we go here, I can say done. Step 2, you can see I've actually, whoever created the protocol has used the component for centrifugation, and there's a timer on it. So I can run the timer as well dynamically in the app here. So you can click run through. At the end of that 3 minutes, a bell will ring. So if I'm running this on a device in the lab, I don't need a separate timer. I can actually use this in the platform to go through. So we'll say done. If I had several quantities, amounts to make a master mix or something, and I've used the amount component to flag these in here, then there's other nice functionality, which is that I can edit the step on the fly. So if I want to change an amount, I can do that. Equally, I can actually scale. So if I had several quantities here of something that I was mixing together and I wanted to make more volume today, I could actually say, well, scale this up for me fourfold, and I'll make more today here. And if I click save, you can see very clearly in red what I've changed, and that's what I'm doing today. And then you can say done. And all of these changes or what I've done and when I've done them is being tracked and recorded in a run record that is getting created as I go in my file manager. So you can see that's already started to be created December 8th today. You can also see it's grey because it's incomplete. So if I close the run record I'm going to go and have lunch, or if my laptop decides to crash or something happens, it doesn't matter. It will save where I was up to. And if I want to, at any time, I can come back and I can resume this here. Another member of my workspace who has access could also resume. So if something happened that was urgently called away, but I really desperately needed someone to finish my experiment for me, then they could resume that record and they'd know what I'd done to that point. And then right at the end, when you've done everything, you would say finish run. Um, After you click finish run, it becomes uneditable. So we'll say that is now, and we'll do a quick refresh in the file manager. And what was grey before is now green, and that's the completed run record. So I now can't edit this, but what I have is a complete record of all the things I did on this day in this experiment that I would be able to correlate subsequently with any data I've generated today in this experimental run. So we have, if you're producing microscopy images or you're trying a different sample approach or something, you'll be able to see what worked and what didn't if you look at the results you get and can correlate back to what you've done on the protocol. So that's kind of using publicly available content. How about making your own protocols? There's a few easy ways to do that. So when you're in, I'm trying to move things on my desktop so I can see what I'm doing. When you're in the file manager, you can see new at the top. So you click there, very easy, make a new protocol, make a collection. A collection, I showed you that paper linked to a collection, and in that you can add multiple protocols. So you'd still need to make all the separate protocols, but then you can put them all into a single collection if you want. If you already have a lot of protocols as Word files or a PDF and you'd like to get them onto protocols.io, there is a bit of a cheat. It's not perfect, but there's a way to import from a file using an AI method that we've added to the platform. So you can upload a file here and it will convert that into protocols.io format. So I did one of these earlier. So I had a protocol that was a Google Doc. She just turned it into a PDF and I added it to the platform. Sorry, the Zoom box keeps dropping down over the top of what I'm trying to show. When I added it into, when I said I wanted to import the protocol and upload it here, it kind of says it's uploaded and then you say convert. And when you say convert, it shows you this protocolify box in the second left, bottom left, sorry. And it's told me here that it completed in 2 minutes. It will track the progress when you actually do it on the fly, but that has actually imported this protocol for introducing fluorescently labeled CRISPR, and we can say view the protocol. So we can have a quick look at that. Oh, it's opening it out. Hang on, it's opened it in a separate box, so I'll open it in here and we'll have a look at it in here. So in the Google Doc, you can see it had title, authors, abstract, before start, and then steps 1 through 16. I have not actually looked at what it did with the import, but you can see, oh, we've got steps 1 through 16, so they've been added into all the separate steps. And if I view the protocol, we've also got the abstract in there and before you start information and the author list that has been added along with their affiliation. So all of that has been pulled out of that file and added into the protocol and protocols.io. So that can save you a lot of time. What it doesn't do is add the components for you. And those were the parts I showed you, like the timer or the content, the quantity. But so once you've got it in, you've got a fast track though to do that. So if you select there, we've got 18 microliters and we come across, we can look for the amount component here and that will add that here like this. So it knows I want 18 and that it's microliter. Do always recommend keeping check because it's not fallible. It will occasionally make mistakes, but you can change the units, you can change the amounts, you can provide additional information if you want to. You can add reagents this way. We've got the timer as well. So if we've got 5 minutes here, we can use duration. And now we've got 5 minutes here like this as well. When you add the timer, it will also add, ask if you want to add to update the timer of the whole protocol. So you say yes, and it's tracking time on the right for the entire protocol. So maybe you want to manually assign how long other steps will take, and you'll get an overview of the complete duration at the top if you do it this way. You can see there's lots of other components, more than I can go through today, but it gives you a bit of a flavour of what's in there. And there's other places. So if you use that tool to upload things, if you want to, you can add the original file if you want, so it's there for comparison. There's all sorts of other information you can add. You maybe think you should have an ethics warning, ethics statement, excuse me, or perhaps there's security warnings you've not got on the original doc. You can add all kinds of other things into the protocol. And if you've got images, you can also upload those. So there's when you're in the editor, you can add image or a video, or you can embed a video if you've got a much larger video and it's on YouTube or Vimeo, you can use embed to add content in that way. So you can much further enhance how useful the protocol is for someone else to follow by adding additional things like images and so on, which we would always recommend doing, really. The file manager, again, I mentioned the other way to create a new protocol is just to click new protocol. So if you do that, it will make for you an untitled protocol. This is what it will look like when you first start. But I really just want to show this to show how easy it is to use. It's literally clicking into the various places and typing. So this is my first section. You can see that table of contents for section 1 has now been created. So it very quickly knows and adds these things as you go along. So you've got that table of contents is happening. Most of the things are happening automatically for you. There's a lot of functionality to add steps. You can do that from the bottom, you can do that from the left-hand side, or sections, steps. You can have sub-steps too. So if something has, if you're repeat wash or something like this multiple times, you can make sure you capture all of it so that if you use that run functionality, you've got all the dynamic elements in there to go through. When you're happy with your protocol, I showed earlier that publish process, that's all up here. It's very easy again. Whoop, my computer's telling me things are going to update. I hope not just yet. The publish process is very easy. There's that process I talked about, and it will force you to fill some things in. So it might say you must include an abstract. So you need to make sure you just complete the pieces that are required, and it will take you all the way through to confirmation at the end. So it's very, very easy to use. I think that's most of the things I was going to show on here. Actually, let me go back and show how you can share with a colleague if you want to. So from this editor view, if you want to share with your group leader or just another colleague, you can very easily click share, and you can add individuals here or a workspace. And you say we can add a person. So Lenny is my boss at protocols.io. So I can just add him. Oops. Sorry, it's doing something funny. If I add him here, I can also change what permissions he has. So maybe I know he's going to finish it and I need him to be able to publish it for me later on. I can give him that permission, or we can just give preview visibility and so on. There's all the different kinds of permissions you can add over here. You do need to click share afterwards, but I'll take him off for now. But that's very easy. Equally, if you just want links for a manuscript, you can go here to get a private link for reviewers and editors, get link, or a blind review link will remove all the author information as well. But you will also get that if you use the reserve DOI process that I mentioned previously, which is very similar to the publish process, which is here. So if you complete that going all the way through, then when you get to the end, you'll have the DOI that would be reserved for your protocol and a reviewer link that way. I feel like I have shared a lot. Let me pop back maybe to my slides momentarily and see if there were other things in there that I should cover. So let me just switch again. Bit confusing, chopping and changing. So just a nice couple of examples of users who've given really nice kind of quotes about how it's been useful to them. So Dr. John Barrows, who was a postdoc, he had said that by putting his content publicly on protocols.io, this has been something that has allowed people to identify him as an expert with the technique. And also thought it looked very good on the CV. So also, if you do publish content on protocols.io and you link to your ORCID or ORCID, then the published protocols will show up under your works on that profile. So they can be automatically pulled across as well. Another researcher, Professor Martellotto, he was a part of the Human Cell Atlas, which is a large initiative funded by Chan Zuckerberg consortium, and through work he's done sharing protocols on protocols.io, he has stated that he thinks probably 80% of his recent collaborations have happened because people have seen his protocols on protocols.io and understood that he's an expert in this area and wanted to work with him. So it can be very beneficial to share your content this way. The examples I've shown you today have been very kind of biological wet lab methods, but the platform is used across all research disciplines. So there are a few example, just links to public protocols here on the platform that cover very different research disciplines. So just to show a little bit how diverse and versatile it can be, you will get a link to the slides, so you'll be able to explore those if you want to see how other researchers have used the platform in different ways as well. Essentially, though, anything that has a step-by-step process, you can add it onto protocols.io. And we mentioned before, lots of different journals and publishers recommend protocols.io during manuscript submission. We do have, I didn't go into detail, but on the publish, next to the publish buttons, there's also peer review options. And from there, you can see we have some closer relationships with some journals than others, where they're much more strongly recommending using protocols.io. But actually, if you look at the author guidelines from most journals, from most publishers, you will see that they recommend sharing full comprehensive methods details, and most of them will probably recommend using protocols.io. Some funders as well, to different degrees, are also recommending protocols.io in their guidelines and their policies. And possibly sometimes when you're writing grants alongside data management plan, you may now be required to add also a method management plan, how you will be sharing methods and making methods available. As I mentioned, quite a few institutions have licenses, so there's an easy way to check for that. A whole host of different places you can come for additional help and support. We have links to past webinars, so if you can't sign up for another one but you'd like to see them more advanced, you'll find recordings of past webinars very easily. And there are tutorials, lots of short little snippet videos that show you how to do specific things on the platform. And our help center does have a lot of information too, but if you can't find it, the information you need, it's very easy to submit feedback or ask questions in protocols.io, or, as I mentioned at the start, sign up for a short demo with one of the team if you want to. And I will stop there, but if you have a moment and are happy to, the QR code here or the URL that will be in the slides when I send it around, and in a moment I'll copy it into the chat. If you can give a little bit of feedback on the webinar, that would be great. I will now also, though, stop the share, and I can take any more questions that anybody has. And let me copy that link out while I'm doing that. And then we can see if you have any questions. If somebody wants to be unmuted, I can do that as well. And I'm just going to come back to the chat. Okay, so I'm popping the link in for feedback, and then I will check the question. So what is the difference between public and private protocols? Does it mean that I can only create 2 protocols in the free version? I can create any amount of protocols, but they're public. Okay, so Perhaps I wasn't completely clear, so apologies for that. When you have the free account, you can make two private files. If you publish one, so it becomes publicly available, so you follow that published process, it will then have a DOI and will be available under a Creative Commons license, openly accessible by everybody. But then you've only got one private file, so you'd be able to make another separate private file. So in theory, if you iterate on content and you're constantly making things public, you can continue to create new private protocols, but you just need to publish them before you'd be allowed to have another private one. Hopefully that's clear. It's to incentivize open sharing, essentially, to do it that way. Great. I looked in the chat, but I didn't actually look in the Q&A. I'm not sure if I don't see anything there. We have finished a bit before time. It's been a while since I'd given one of these, so maybe I went fast. And make sure I showed everything I intended to in the demo, but feel free to add questions as well. Would it be possible to get information on more components, how they can be used later? It's very laborious to add when adding several protocols. Like to have a clear view of how they can be used later in order to decide which one. That's a really good question. The primary two that can be used, really, I showed you, which is either duration or things like centrifugation, where you can add a timer, and the amounts, the volume. They're the ones that can really be scaled more in the platform. However, that said, let me share again, actually. Some of the other things like the reagents and so on can be really valuable. Not sure where I am here. Oh, I think I think my Chrome is trying to do an update and it's just shut itself down. Let me try and go back in and share it again. I'll try and find a protocol. So when you add in things like components, I think that's what my message was about earlier, that it was going to shut down, that my Chrome wanted to do a restart. So I'm going to have to come back in. It's getting all sorts of flag notifications from my platform. Now we have to wait for my computer to reload. Reagents will automatically be pulled, or equipment will automatically be pulled into, nothing's loading, is it? Into the materials tab. So you can automatically create essentially your list of materials and equipment if you use that. Stop the share and see if I can get something up because it's not loading right now. So reagents and equipment are quite good to use that way. And software as well, because it will give you the links. And if you are doing something computational and you use command or data set, then you can add in kind of specific links to those things. And if you've got snippets of code that you add as command, it will facilitate copy paste of those very detailed commands or little pieces of code sometimes, which will mean you can run through a computational workflow much more easily. There are others. I think the advanced webinar might help with this too. There is a smart component. Sorry, nothing is loading in my in my. Let me see if I can get a different browser up. Safari, in my Chrome at the moment while I'm doing this. There's a smart component which allows you to have a key value pair. So you can define in a smart component. I think I can get back to here we go. Let me share this instead. This will help show. I can actually talk it through as I go, using Safari instead of Chrome. So if we're in and we go to components and use smart component, we can add a key value pair. So we might put number of cells and we might require an answer, and we might put percent dead cells or something like that and require an answer. And if we add that component, then when you do a run of the protocol, you'll be required to input values into the values for both of these as you go through. So that component can be quite helpful if you want to prompt for observations during a protocol run. Um, Go to allows you to kind of point to an earlier step if you want to. I mentioned command that will allow you to kind of define specific snippets of code and so on. Um, Protocol allows you to link to a sub-protocol. So if you're running through a big experiment and there's some kind of modular piece that you might do multiple times, like you might have the buffer recipe. If you need to create more buffer, follow this recipe and point to that other protocol. You can do that way. So you can nest in sub-protocols. You can only have a layer of two though, so you can't do kind of other weird quirks beyond that, but it will allow you to nest inside another protocol. Um, Notes and things just are kind of for visibility. Um, so you can add important info. You can have those be collapsed in the view mode or have them open out similarly to that, but in red, safety information we have here. There are keyboard shortcuts, which has just prompted me there. So you can do things and shortcut through them there. You can cite papers as well. So you can search PubMed, search public protocols or protocols, and or just create a citation. So if you want to cite a paper in the middle of the protocol or anywhere else, actually in the description or before start, like put other citations in, you can search using content here. So if I looked for Ganley, I'm going to find my husband's publications, but I could pick one of his papers um, and cite that in, and it's going to add that citation into the text for me. And that will also auto-populate under the reference list if we view it. You can manually create that reference list as well, but you can also use the citation manager to do that. Oh, things are going slow, I think, because I'm sharing and zooming at the same time later. So here now that reference is added and it tells me where the reference is in there. So you can see kind of it will create things on the fly for you as you go through if you use some of them. So if I used the reagents one to add in specific reagent information, that would auto-populate under materials I mentioned, but I didn't show you earlier. So there's quite a lot of places you can do that. I'll go to the next question. Can you comment on the integration of protocols.io with ELNs like RSpace? Yes, I can. So we do have a few different integrations in protocols.io. When you are in your folder, it's a bit easier to show here. If you want to integrate and kind of be able to export or import content to a different location, we have export, and if we press on JSON here, oh no, that's via the API. Excuse me, that's not the right one. If we go to PDF, it will give add apps as an option. So if we click on add apps, it will show you the various different integrations that we have. So some are more kind of storage locations like Box and Google Drive. Maybe your lab works with a specific resource, OneDrive, OSF. Some universities have a subscription with OSF and have space there for content. Some are more specifically electronic lab notebooks like Lab Archives, Lab Forward, SciSure, and sometimes they're on the other side. So these are ones where you can push to that electronic lab notebook or that file location. RSpace, the one that you've asked about, and I think SciNote, they have integrations from the electronic lab notebook. So from being logged in on the electronic lab notebook, you can log into protocols.io and pull content out. So they work that way. You might be able to push to, I'm not 100% sure, but it's more configured by and controlled by the electronic lab notebooks. If we hear about other integrations that people might like, then we can try and add them. But these are the ones that we have right now. Hopefully they're useful. Let me stop the share again. And I mean, we've got a few minutes left, so I can hang on. But if people don't have any more questions, we can stop there. And people are very welcome to email any questions after the fact. My easiest email to get me directly is emma@protocols.io. Very happy to take questions later, or if you're thinking about it later on, and anything comes up, let me know. I'll stop the recording, but I'm still here for a couple more minutes. So if someone's typing, I don't want to cut you off. But we'll stop that.